Integumentary system basically relates with the skin. You can also talk about the glands associated with skin. And in the skin, we will have a skin epithelium, which is epidermis. So, epidermis is the skin epithelium, which we understand it will be keratinized because skin is kept dry. It is keratinized, stratified squamous epithelium. You need multi-layered epithelium because it is resistant to abrasion. So, you want resistance to abrasion because skin is under constant abrasion. Then there will be dermis. Below that is hypodermis. Hypodermis is basically attaching the skin to the deeper tissue, and there will be some fat included, fat tissue included here. But then dermis, if you are talking about dermis, there will be superficial papillary layer of dermis, superficial papillary layer of dermis, and then there is a deep reticular layer. If you draw the skin in a diagram, like this is the epithelium, the epidermis, and this will be the superficial papillary layer, loose irregular connective tissue, and this will be the deeper reticular layer. The collagen fibers are present running north, east, west, south. So, we understand dense but irregular connective tissue comes as a question. I will repeat. When you are talking about integumentary system, you have to look at the skin and the glands. In the skin, we have to talk about skin epithelium, which is also called epidermis. It is having keratin to keep it dry. So, keratinized stratified squamous epithelium. If we are looking at the dermis, dermis will be having two components, the thinner one, superficial papillary layer of dermis, and thicker one, which is deep reticular layer of dermis, which is having dense but irregular connective tissue because collagen fibers running north, east, west, south, not arranged regularly, going in all directions. So, irregular, dense irregular connective tissue. Whereas superficial papillary layer, you have loose irregular connective tissue. Now, as we proceed further, the epidermis, the skin epithelium which we are talking about, can have 5 layers if it is thick skin, say for example from the palm and sole, or if it is from thin skin, then one layer will be missing, the second layer stratum lucidum may be missing. So, let us look at the 5 layers of skin in a diagram. We will draw and then look at the histology specimen also. So, if we are telling that this is the skin epithelium, the very first layer you will find, number 1, stratum corneum. Stratum corneum will have keratin fibers. So, keratin fibers are required because to keep the surface dry. This is number 1. Number 2, you will find stratum lucidum. Stratum lucidum is having a thin homogeneous pinkish appearance, eosinophilic, takes more of eosin. These layers are dead cells basically, these are dead cells, or you can also say acellular. There are no cells. Cells are there, but they are dead and their fragments are there, they are getting desquamated because the superficial layers get desquamated. Now, when you look at the layer number 3, which is stratum granulosum, you will find that there are cells which are having granules, they are keratohyalin granules, and these keratohyalin granules are important because they will help in formation of the keratin fibers. This is layer number 3. Layer number 4 is quite large and it will have some cells which are, if you prepare the histology slide, they will appear spiny ends. So, why they are having this spiny processes? Because there were desmosomes holding these cells together, like here and here. This is one desmosome holding the cells with each other, but if you prepare the slide, they detach from each other. So, those desmosomal ends basically they are appearing spiny. That is why it is called stratum spinosum also, stratum spinosum. Spiny cells, they are keratinocytes basically. And here we have made a specimen, that is why desmosomes are broken, but sometime it may happen autoimmune disorder like pemphigus vulgaris. In pemphigus vulgaris, you may see that there was autoantibodies destroying the calcium cadherins, which are the cell adhesion molecules for the desmosomes. So desmosomes were separating. There was acantholysis, and it is intraepidermal lesion, and this intraepidermal lesion is producing skin blisters. Skin blisters are seen in pemphigus vulgaris. I'll repeat, it involves this layer number 4, which is quite thick. In the layer number 4, you have these cells which are having spiny processes attached by desmosomes. So normally these desmosomes keep holding cells together in stratum spinosum, but it may happen that there is an autoimmune disorder, pemphigus vulgaris, autoantibodies against cadherins, which is cell adhesion molecule for desmosome, destroyed. So acantholysis, the cells are separating, there is intraepidermal lesion, and skin blisters will come. These are all MCQs. Now look at the last layer, which is layer number 5, that is stratum basale, because it is at the base, or you can say stratum germinativum, because it will have the stem cells which will keep germinating the rest of the. So stem cells are there, and these stem cells, they keep dividing and keep adding more and more layers, so that because superficial layers are anyway going to be desquamated. Now in the stratum basal or stratum germinativum, not only you have stem cells, you have other cells also, like one you can talk about is melanocyte. So melanocyte is present for giving the pigment melanin in the stratum basal, though its processes can go into stratum spinosum, but cell is in the basal layer. And in the basal layer you also have another cell, which is the Merkel cell. Actually Merkel is a receptor for light touch sensation, like if you are talking about holding a pen in your fingers, sustained touch, that is detected by Merkel receptor. So these are the 5 layers, as we are telling: stratum corneum, lucidum, granulosum, spinosum, and stratum basal in thick skin of palm and sole, if you have taken the specimen from palm or sole. But if you are talking about the back of the hand, thin skin, then second layer will be missing. Stratum lucidum is missing in the thin skin. Now let us look at a diagram also, schematic diagram and histological diagram, to identify the structures. You have to understand that this all is keratin. This all is keratin. So stratum corneum, which can be shown here also, this is all keratin. So this is stratum corneum, the first layer, dead acellular layer. Number 2, you can see stratum lucidum also, thin homogeneous structure, as is evident here, but it is missing from this specimen. Here you don't have it. That means this is taken from a thin skin like back of the hand, and this is taken from thick skin, maybe the palm or sole, because the second layer is evident here and second layer is missing. Straight away you are seeing the third layer, which is stratum granulosum. So how do you know it is stratum granulosum? Because it is having these keratohyalin granules. These are the keratohyalin granules which help in formation of keratin. So here these cells you will find, if you magnify, you will find they'll have these keratohyalin granules. This is the third layer, this one, this is the third layer. Otherwise, if you are looking at most of the skin here, starting from here, this area, this is all stratum spinosum, except the basal layer, it is all stratum spinosum. So this is the stratum spinosum. You just have to remove one basal layer, rest all is stratum spinosum, the largest of all. The same can be seen here from this level to this level. You just have to exclude the basal layer. So this is the stratum spinosum. Here you see one additional cell, which is Langerhans cell. Langerhans cell is antigen presenting cell, and it will pick up the antigen from the skin and give to the lymph nodes. It can be compromised in conditions like histiocytosis, more of Langerhans cells then. So this is the Langerhans cell, and it is dendritic. It has dendritic processes, and it is antigen presenting cell in the stratum spinosum. We also told that these cells are held together by what is called as desmosomes, and if these desmosomes are broken, then you can, pemphigus vulgaris is the problem where you will have skin blisters. So desmosomal problems, autoantibodies against the cadherin of desmosomes, and intraepidermal blisters, pemphigus vulgaris. Now look at the basal layer. This is the basal layer, and that is shown here also. Stratum basale, or you can say stratum germinativum, because here you have the stem cells. You see mitotic activity here. So number one. Number two, you can see melanocyte. Melanocyte is present to giving the pigment to tackle the ultraviolet rays of the sun, present in the basal layer, but processes are moving into the stratum spinosum also. And this is the Merkel cell we were talking about. It is at the basal layer. It has this nerve fiber also, and it is to detect the light touch sensation. Light touch sensation, which is sustained like holding the pen, is detected by Merkel cell. Now we have to talk about appendages like hair and glands as well. When you talk about the skin glands, you can tell that there will be the holocrine gland. Holocrine means entire of the cell gets damaged and becomes the secretion. The sebaceous gland. So sebaceous gland, we'll see a diagram also, is a holocrine gland. Entire cell gets broken up. Or it could be acrine gland, which is the sweat gland. Sweat glands are two types, acrine and apocrine. Mostly they are acrine. Here, no cell membrane loss. No cell membrane loss. The cell is intact, only secretions go out. And the secretions of acrine gland are thin and watery, like you see the actual sweat. And when you talk about the third one, it is the apocrine. Apocrine sweat glands are rare, and apocrine sweat glands, as the name is suggesting, the apical portion of cell membrane is lost. Cell membrane is lost, and it is covering the secretions basically. And apocrine glands are basically rare, as we told, can be in the axilla, can be around the nipple, perinipple area, can be in the genital area, perineum area, around the urethra, vagina, anal canal, where you have hair. It relates with hair because the secretions move into hair follicles, and the secretions are thick and viscous. Thick and viscous. This is sweat gland. So sweat gland is the only gland which is sympathetic. Otherwise, all the glands are parasympathetic cholinergic. All glands are parasympathetic and cholinergic. Only one exception, that is sweat gland, because sweat gland is sympathetic. S for S, sweat is sympathetic. Now sympathetic also, it is sympathetic cholinergic or sympathetic adrenergic. If you're talking about sympathetic cholinergic, it is the acrine variety which we have discussed. It is more common. The sympathetic adrenergic is apocrine variety, which is less common, like we saw in axilla, perigenital or perinipple area. So these are some of the questions which keep coming, we wanted to take care of. Also, you can look at the diagram here. This is the skin epithelium, keratinized, stratified squamous, because multiple layered, and keratin is also seen. This is the hair follicle we are looking at. Related with the hair follicle, you will see this is the sebaceous gland. That is how the sebaceous gland appear. The peripheral cells are intact, but as you go towards the hair follicle, they are getting totally destroyed. Whole of the cell is destroyed. So holocrine, this is holocrine sebaceous gland, giving the sebum into the hair follicle, which will lubricate the hair follicle and skin also. So secretions go along with the hair follicle like this. Also, if you are talking about apocrine sweat glands, they are deeper in dermis. Apocrine sweat gland, they are deeper in dermis, and even their secretions go into the hair follicle only. thick and viscous secretion under sympathetic adrenergic pathway. This is sympathetic adrenergic pathway and thick and viscous secretion, and they are deeper in dermis compared with the eccrine gland, which is superficial and it is sympathetic but cholinergic. Sympathetic cholinergic give thin watery secretion, thin watery secretion, and the secretions go directly to the surface of the skin. They don't open into hair follicle. So this is about the eccrine, which is also called as merocrine. Merocrine is a journal term and no loss of cell membrane, basically only the secretions are given out. Here you can see some muscle also, which is erector pylorum, smooth muscle. This is erector pylorum, the smooth muscle, and this is to erect the hair follicle under sympathetic system. Fight and fright, goosebumps, that is done by erector pylorum under sympathetic system. Fight and flight, emergency situation, and here you will see the hair is erected, goosebumps, that is what we are talking about. In the skin, we have told there is skin epithelium, the epidermis. Otherwise, there is this loose areolar connective tissue, which is superficial papillary layer of dermis. Otherwise, there is deep reticular layer of dermis. So deep is reticular layer. Reticular layer of dermis, we have told that it will be dense but irregular connective tissue, deeper. So this is about the structure. In tegumentary system, skin, the glands and the hair follicle.